Hey, listeners of the Bio Report. Before we get started this week, I wanted to tell you about the digital library from Deep Dive. How much time does your team spend looking for research papers? Google, PubMed, social media. There's got to be a better way. You can now search a reference database of 100 million scientific papers and read the full text of 20 million articles, annotate them, and share with colleagues. It's the smarter way to do research. Here's the best part. If you're like me and been frustrated by not being able to access articles you find because they're behind a paywall, I've got good news. With Deep Dive, you get one-stop affordable research. If you're a listener of the Bio Report, you can try the enterprise version of the service for free for 1 month. Go to deepdive.com/podcast and enter the code BioReport. That's Deep Dive, DeepDive.com/podcast, and the code is BioReport, one word, all caps. I'm Daniel Levine, and this is the Bio Report. The marriage of synthetic biology to mRNA is creating the potential for a range of programmable therapeutics that can provide new ways of treating deadly and chronic diseases. Strand Therapeutics is creating a platform for developing these long-acting mRNA drugs that it says can be precise, multifunctional, and deliver potentially curative treatments with a single dose. We spoke to Jake Becraft, co-founder and CEO of Strand, about the company's programmable RNA therapeutic platform, how it works, and the indications it's pursuing. Jake, thanks for joining us.
Thanks for having me here.
We're going to talk about mRNA, Strand Therapeutics, and its efforts to develop the next generation of messenger RNA therapies. mRNA as a technology became front and center in the public imagination with the vaccines for COVID-19. My guess is, though, that most people don't understand the basic technology or its potential beyond vaccines. Perhaps we can begin with mRNA itself. What is it, and what makes it compelling as the basics for therapeutics?
It's a fantastic question and one I'm happy to answer. So there's something in biology that Jim Watson of Watson and Crick, people probably remember from biology class, Jim Watson called the central dogma. And the central dogma is essentially that DNA, which a lot of people know and remember learning about in school, DNA makes messenger RNA. Messenger RNA makes proteins. Proteins are really the essence of life. Proteins are everything that makes up what you think of as your physical being. And we can make drugs out of proteins as well. We've all sorts of things like antibody drugs, things like insulin, are proteins that we can create synthetically and then inject them into the body. Why mRNA is such a breakthrough is that what mRNA really is an information molecule. So you imagine that your cells contain DNA and they are the source code of your being. And then you have proteins on the other side, which are everything that makes up your physical personage. And in between, that DNA needs to send a message to your body to make more proteins. And in order to send that message, your DNA wants to continue to be in the nucleus, continue to be protected because you don't want to damage kind of the source code. And so it sends these messages out into its cells. And that is in the form of this information molecule that we call messenger RNA.
You're a synthetic biologist by training. I think when people hear synthetic biologists talk about developing a therapy, they can sound more like engineers than biologists. One other concept I'd like you to explain is gene circuits. What's meant by that term?
So that's another fantastic question. So what we're the way that your body produces all these proteins that I was just talking about, you have a number of genes, and those genes on your DNA encode for proteins. The thing about your body is that those genes are not always on. Really, everyone knows about how your stem cells differentiate into different types of cells, right? You have the same DNA inside of your stomach as you do inside of your skin cells. And so the way that your body is able to remember the type of cells or go down these different differentiation pathways is through something that is called is often referred to as a circuit. It's essentially the way that your body turns on and off different genes in your body. With synthetic biology, what we found is that the underlying circuitry that underlies all of life is very similar. Those circuits are very similar to how we think of computer code and how we can encode different sorts of programs through the use of different sorts of circuitry and algorithms. And so, what we're able to do at the highest level is take natural circuitry of how your body processes genetic information and recode that in order to do things that are interesting from a therapeutic standpoint, right? And so I guess the easiest way to think about it is you can think about a therapeutic, an mRNA in Strand's case. So in Strand's case, we build genetic circuits on messenger RNA, and that allows us to deliver messenger RNAs that they're injected into your body, and maybe the point of that messenger RNA is to go into a tumor and actually kill that tumor so that you can be cured of your cancer. And in order to do that, you need to be very safe because you don't want an mRNA that is programmed to kill ending up in your liver, for instance, and telling your liver to kill some of its cells. And so what you do is you build a genetic circuit, and that circuit has three parts. It has a sensor, it has a processor, and it has an output. And so if the output is that the cell kills itself, and that the input could be biosensors that sense for whether or not it's a tumor cell. And so now and then the processing unit is the actual circuit itself. And so what happens is the mRNA goes into these different cells, it goes into your liver, and it goes into the tumor. And the biosensors pick up the what is going on inside of the cell. And then the processing unit, the genetic circuits, make a decision. They say, due to what we're reading on our sensors, this is a cancer cell or it isn't a cancer cell. And if it gets a reading that it is a cancer cell, then the circuit's output becomes, kill this cell. And that can lead to revolutions in the way that we treat disease, from everything from cancer to Parkinson's to cystic fibrosis.
So it's a mind-bending concept, but how good are we at programming today?
Genetic programming has been growing at an unbelievable rate. So right now, I would say we are entering the personal computer age. If we are going to overlay synthetic biology and biological engineering with technological advancement of the late 20th century. So if you think about like the early hacker clubs, things like Wozniak and Jobs met at a hacker club in the 70s before launching Apple. That's about where we're at, right? We're at where we have a passive understanding of how these systems work. And we can start to actually string pieces together, string different elements and circuit components together to make things that are very useful. Now, we're not making iPhones, we're making small personal computers that run on MS-DOS essentially and can handle some basic functions. But just like computers at that stage were, revolutionary in their time to automate certain tasks that were very time consuming, and then led to even more advancements, which lead to, the sorts of crazy levels of technology we have today. That's what the future looks like for synthetic biology and for really the future of biological engineering.
There are many challenges mRNA therapies present. This has to do with how long they may last within the body, challenges of delivering them to where they need to go, ensuring that they don't go where you don't want them to go. How many of these challenges can be addressed through the programming of the genetic circuitry as opposed to other components that make up the therapy?
So mRNA is an incredibly simple molecule and drug product. So if you think about the COVID vaccines for a moment, there's really two components. There's the messenger RNA, which is this information molecule. Your body is filled with messenger RNA as well. It's a very transient molecule. And what I mean by that is it's very fragile. So since it's an information carrier, it's almost like a Snapchat. It's meant to be seen or to be made into a protein and then basically to disappear. And that's actually what makes it such a fantastic paradigm for therapeutics. You're able to send a message to the cells, have the cells create a therapeutic protein that corrects that either corrects something or primes the body's immune system to be able to protect itself against a virus like COVID, and then it goes away and it's done its job. The other component of the drug is just what's called a lipid nanoparticle, and so that's really just a layer of fat. Essentially, a lipid is just a fat that is formed around the messenger RNA, and that just helps protect it because again, it's very fragile. And so, in order to get into the cells, you need to have this protective carrier, and it is taken up into the cells. So you only have those two components to work with, and so when you're thinking about how to solve the issues of specificity, and really it's more with solving the issues of messenger RNA as a therapeutic, it's more about creating longer lived species in the body than shorter lived. The COVID vaccines exist in a patient's body for a day to 2 days at most before they're completely eradicated. Just because your body is constantly turning itself over and it's constantly degrading things so it can continue to produce new things and respond to its environment. And so when you think about how do you change how do you take that technology and now use it to fix something where you need a longer lasting expression, something like enzyme replacement therapy, where someone is deficient in a particular type of protein or enzyme. In order to do that, you really have to innovate on the messenger RNA molecule, because again, there's only two components. There's the messenger RNA and there's the lipid nanoparticle carrier. Gene circuit technology and synthetic biology allows us to both increase the duration of time that the mRNA is able to exist for, so from a few days to maybe a few weeks. It allows us to, achieve higher specificity levels, so that the drugs are even safer. And the technology can really, the sky's the limit in my mind of what we're going to be able to accomplish with it.
As you think about the challenges of moving an experimental therapy into the clinic, where are the biggest hurdles you have to overcome?
So with any drug development procedure, you need to be first very convinced of its safety. So in order to get into patients, you need to be convinced of its safety. But in order to be convinced that you should invest in this drug, you have to be also convinced of its efficacy. And so I think that the big advancement that we're going to see with things like messenger RNA therapies is that every time, since it's a platform, right? Since every time we change the drug, it's still made of the same molecular makeup, right? It's still messenger RNA and a lipid nanoparticle. It actually lowers the bar it lowers that safety bar. And so now, like as Strand Therapeutics is a company, we plan to enter the clinic next year with our first drug. But after that, every time we enter the clinic with a new drug, it brings that safety barrier down, because now we know that the last thing we did was safe, and now we've made a slight change, or we've changed a little bit of what we're going after. And how the FDA will respond to that sort of breakthrough in how we build drugs is still up for debate. I think that by increasing the speed at which we're able to develop drugs, though, we'll be able to also drastically reduce the cost of development And by reducing the cost of development, we then reduce the amount of investment it takes, and it'll lead to both an increase in innovation and a decrease in the cost of drugs.
You mentioned you using nanolipid particles as a vector for these therapies. One of the problems with nanolipid particles is their specificity, their ability to target precisely where you want them to go. How much of a challenge is that? And does the use of a gene circuit allow you to work around that problem?
Absolutely. I mean, that's the value proposition right there. A lipid nanoparticle is it's not necessarily 100% specific. There are different sorts of lipid nanoparticles that you can use that can go to different tissues and different sort of distribution percentages. Right? You can have some that mostly end up in the liver. You can have some that mostly end up in the spleen. You have some that you can make mostly end up in the lung. But at the end of the day, it's a distribution, and it's not overtly specific. And so with the power of gene circuits, you can actually complement those two technologies together. And say if you have a lipid nanoparticle that 70% of your drug goes to the spleen, and say you're targeting the spleen with some sort of a therapeutic, then you can make a genetic circuit that only expresses in the spleen. And by marrying those two technologies together, you now have delivered a sufficient amount of drug to the spleen, but you've reduced the amount of what's called off-target activity by engineering messenger RNAs with gene circuits that prevent them from being active in other tissues such as the liver. That's really the value proposition there. So you bring those two together, and between their distribution and the specificity of the genetic circuits, you can have complete specificity in delivery of the active particle.
When you've got a platform technology with as broad a potential application as you do, to me, one of the biggest challenges is prioritizing indications you're going to pursue. Strand is initially focusing on immuno-oncology. Why start there?
That's also a great question. So where we first started was, where can we have the most impact right now? So the science around immuno-oncology has actually become incredibly robust in recent years. We know that certain proteins, certain protein drugs, things like cytokine therapies, combined with checkpoint inhibitor drugs, for instance, we know that those drugs can be incredibly effective at treating tumors through an immunotherapy approach. The real problem with a lot of these drugs comes from their pharmacokinetics, how the drugs move around the body, how long they last in the body, and then their toxicity. Right, when they start to move, or they're administered into the bloodstream, you have these proteins floating around that are meant to stimulate the immune system. You end up overstimulating immune responses and leading to incredible toxicity and even death in a lot of cases. And so, taking an engineering approach, we thought that the bar was lowest, where we could have the biggest impact immediately in immuno-oncology, and use that also as a way to get our therapeutic into patients, to provide value to those patients, and also to provide value to the company. Because as I mentioned, every time we go into the patients with this therapeutic, we become more and more convinced of its safety. And so that obviously takes an incredible amount of risk off the table. It leads to higher investment for the company, which we can put towards making new therapeutics for more patients.
Strand's not expected to be in the clinic until 2022. As you start thinking about dealing with regulators, how does the experience with vaccines change the regulatory landscape? Do you think, given the broader use of mRNA vaccines with COVID and the fact that there's greater comfort among regulators than there might otherwise have been, makes it an easier discussion?
I absolutely think so. I think there's always the unknown, right? And I think that one thing that's important to drive home, especially when we're thinking about the vaccines and people's thoughts on the safety relative to the fast timeline that these therapeutics were delivered, they should know that the FDA is always errs on the side of caution. And so I think before COVID, there was knowledge that mRNA was a very safe molecule as a drug paradigm, but there wasn't, billions of data points. Now, post vaccine, hopefully we're going to be seeing, we'll have known that, likely the regulators that we'll be talking to directly will have gotten mRNA injections themselves. And so, with that sort of in hand, I think it lowers the bar. I mean, of course, we still need to be incredibly diligent in forecasting our safety, but with a new platform, bringing new platforms forward is always expensive because you need to really convince the FDA and other regulators that what you're doing is safe. And so, having this sort of advancement, having this sort of acceleration of data that will show just how safe these therapeutics are, really brings, I think, the conversation home. And it means that we're having more of a conversation about what we're actually doing with our therapeutics, how we're activating the immune system against cancer, for instance, rather than having to talk all about rather having to also then double our work because we also have to talk about the platform itself.
In January, Strand and Beijing announced an agreement for developing and commercializing your immuno-oncology mRNA treatments for solid tumors. This is a deal for China and certain Asia-Pacific markets that provided you with $5 million upfront and an additional $28 million in near-term milestones, with a total deal value of up to $277 million plus tiered royalties. Why Beijing, and how do you see this deal accelerating your own work?
Yeah, Beijing is a wonderful collaborator. I think that we were looking for a partner that could help our first drug access the Chinese market. So we develop our drug in the United States and in other countries that we're very comfortable, or that we're very knowledgeable in working in, and know the pathway forward for commercialization. But the Chinese market is a different animal. As a global company, I think we want to be able to get our drug into as many people as possible. And so Beijing presented an opportunity to both, capture deal value. You mentioned kind of some of the economics of that deal, which I thought was, which were, incredible accelerators for our R&D pipeline as a company, getting that money into the company at such an early stage. We were less than 2 years old at the time that deal was struck. And having that money in-house really accelerated those efforts. And then further than that, it led to this collaboration with a group that is highly experienced in oncology drug development. And so being able to, sit across the table from people who have put, a double-digit number of oncology drugs into clinical trials and be able to talk to them about, what the strategy of different clinical development is, those are sorts of resources that an early stage company just doesn't have. And being able to work with someone like Beijing, who can help us access new markets as well as help us, oversee some of our strategy, is incredibly valuable for an early stage company. And and then the economics are really just the cherry on top.
Beyond immunotherapy, how do you see the potential for what you're doing being able to reconceive the idea of gene therapy?
So the idea of gene therapy, and I define gene therapy pretty broadly, right? It's the idea that you can take a gene and add it to a patient in some sort of a safe manner, but in a way that it helps with a disease, either with the protection or the treatment of a disease. And so messenger RNA as a gene therapy platform is incredibly interesting, really because it's temporary. I think that messenger RNA going in and fixing different sorts of diseases that may arise is just so incredibly. It's just an incredible breakthrough opportunity to not get into the realm of genome engineering, which I think for some diseases is also coming. Things like hemophilia and such deep genetic diseases that need to be fixed at the genetic level. We can focus on there with other sorts of technologies like CRISPR. But messenger RNA allows us the opportunity to take temporary approaches which could have, permanent solutions to diseases that completely ruin people's lives. And things like developmental disease, spinal muscular atrophy being one of those diseases that I think is just awful. Other diseases like that can be treated with messenger RNA in a way that doesn't require actual genome modification in any way whatsoever, but still leads to a lasting curative effect.
Jake Becraft, CEO of Strand Therapeutics. Jake, thanks so much for your time today.
Thanks for having me.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.